Okay, thank you very much, actually. Okay, so I realize this is a 2-hour session this afternoon, but I think we're all going to want to have a break at around halfway through it or so. So I kind of split it into kind of 2 halves and finish, probably kind of take about 50 minutes for the first half and then 50 minutes for the second half so that we can have a 10-minute break in between the two. Okay, so what we're going to look at today. So yesterday, basically, what I tried to do is kind of really give a brief outline and a brief history of various sort of continuous level models to describe biological movement processes. And we kind of talked about kind of reaction diffusion models, and reaction diffusion models obviously have a kind of a diffusive element. And, in classic sort of mass conservation arguments, you can kind of define some sort of Fickian diffusion type term where the population moves down its own gradients. I described chemotaxis, or generally taxis type models. So those for kind of describing the movement of a cell or an organism according to some chemoattractant. And in those kind of chemotaxis type models, you basically assume a kind of a taxis type flux where the population moves up the gradient of some attractant Q, call it V here. And then I kind of very briefly kind of mentioned at the end of yesterday's talk some non-local PDE models that are used to describe processes such as cell adhesion or swarming type behavior. And they kind of incorporate non-local elements into the model. But again, essentially, they can be derived from these kind of classic conservation type arguments where you describe some sort of flux of the population according to the distribution of the population in the surrounding region. So, as I say, there's been a lot of kind of models for biological movement formulated at this continuous level based on kind of logical arguments about the movement of the population. So that was kind of the main thrust of yesterday's lecture. But where I kind of left it at the very end of yesterday's was kind of talk a little bit about, that's all very well at a kind of a population level, but what happens when we start to look down at the level of an individual and actually how they respond to their environment? So kind of taking the example of kind of chemotaxis, then the E. coli bacteria cells that I talked about yesterday, these are very small rod type bacteria. They're really only a couple of microns or so in length, basically. And at that kind of length scale, they're really too small to be able to detect an actual gradient of a chemical across their body length. Very small fluctuations in the environment. You can't detect a concentration gradient at that scale. So they simply respond to the concentration that they observe. Dictyostelium cells and other larger cells are, of course, that much larger, basically. As I said, they're kind of 10 to 20 microns. And at that kind of scale, then you've actually got the capacity to work out a spatial gradient that you compute across your body length. So there's a difference then between the way cells such as E. coli and cells such as Dictyostelium are responding to the environment. And that, of course, also kind of moves up to the larger scale of organisms. If you take certain species, such as snakes, then snakes have, of course, evolved these very forked tongues. And those forked tongues are incredibly important for their sensory capacity, that kind of sort of separation of the tongue and having receptors at the end of each of those tips allows them to effectively compute local gradients in substances that they're detecting in the environment. Okay, so at an individual scale, we've got lots of different ways of responding to the environment. So in other words, there's more than one way to sense a concentration or sense a gradient. And kind of coming back to this whole sort of problem of chemotaxis, this kind of what chemotaxis is actually, you can kind of get a sense of a little bit of kind of a confusion just by looking up its definition in various dictionaries. If you go to sort of like the Oxford English Dictionary, they're kind of an online source, then you actually read quite a precise definition: the oriented or directional movement of a motile cell or organism in response to a gradient of concentration of a particular substance. So it's a quite specific definition there, talking about movement in response to a gradient. If you kind of look at a kind of another kind of very well-known dictionary, Collins, it simply says the movement of a microorganism or cell in response to a chemical stimulus. So this kind of whole definition of chemotaxis, you know, it's got little different sort of subtleties in terms of the way it's phrased. But when you think about the macroscopic level, whether or not you're responding to a gradient or responding to simply a concentration, you can still see similar macroscopic phenomena. These E. coli cells that are only able to detect a concentration, well, if you kind of have that kind of cue coming out of a pipette, then you're able to get the aggregation of the cells near that attractant source. And similarly, those dictyostelium cells that have the extra ability to detect a gradient, again, they will accumulate near the source of the gradient. So even though we've got a lot of distinctions at the kind of the microscopic scale, at a macroscopic scale, we're seeing similar sort of behavior. Okay, so let's kind of go a little bit, again, a bit of a background here, actually. And as I say, you know, chemotaxis was first studied back in the late part of the 19th century. It was studied intensely over that period. But then as people started to get a little bit more understanding of the way organisms were responding to the environment, it became clear that they needed a slightly more precise terminology. And the one that's very much still used today is that due to Frankel and Gunn in terminology. And essentially, they describe two principal types of movement. The first one is what we call a kinesis. And in a kinesis, then we have a change of movement according to a signal intensity. That's like my, that's effectively my E. coli cells. They detect the concentration, the signal intensity, and they change their movement accordingly. But then within that kind of broader kind of level, we can actually still think about even further subtleties in terms of the way you do that, because of course, according to a concentration, you could change different parts of your behavior. You could change, for example, your speed, or you could change your turning frequency. So again, there's extra subtlety. In orthokinesis, the intent is where the intensity will alter your speed. Clinokinesis is where the intensity will alter your turning frequency. So that's how a kinesis was described. And then a taxis is effectively a change of movement according to a signal gradient. So being able to detect a spatial gradient in some substance. And again, that has further sub-definitions, tropotaxis, where you're able to instantaneously calculate that spatial gradient. That would be like my DDs, dictyostelium cells, that compute the spatial gradient across their surface. But then there's other kind of taxis, such as clinotaxis, where you indirectly calculate a gradient. In other words, basically, you calculate the concentration at one point in time, at one point in space. You move somewhere else, you calculate the concentration there. So you're effectively calculating the concentration at two space points and two time points, and then you compute the spatial gradient from that. Okay, so there's these various terminologies, and they each imply different sort of capacities of the organism. Something like orthokinesis, that can be achieved even with just one receptor, basically. You detect the concentration and you change behavior. Clinokinesis, again, would just require a minimum of just one receptor. A tropotaxis would require at least two receptors to be able to compute that spatial gradient. Clinotaxis, well, actually, you would only need one receptor, but potentially you have to have a memory as well in order to kind of recall the concentration from that previous time. But essentially, as I say, we've got these various ways of defining the response of an individual to some sort of chemical substance at the individual level. So, just to give a couple of examples on that, basically, that E. coli that I talked about earlier, they can't do taxis. Their response to the chemical is a clinokinesis one. They basically change their turning frequency in response to the concentration that they're perceiving. If you take something like nematode worms, which is a very small organism, it's a multicellular organism, but it's still a very kind of small sort of scale, but they also have a very strong chemotaxis response, and their movement actually combines some of these things. They perform both clinokinesis and clinotaxis. So, and those are just done a note on this. E. coli is the most intensely studied organism for its chemotaxis behavior. But of course, actually, it doesn't do chemotaxis by this terminology of Frenkel and Gunn. It does clinokinesis, but it's still, if you go into the literature, they're always talking about E. coli chemotaxis. And this is really very much a historical thing that dates right back to the very early stages of when chemotaxis was first uncovered in the 19th century. Okay, so we've got all of these different ways. But does this really matter then at the macroscopic level? So whichever of those kind of four schemes you kind of take, whether or not you kind of do a clinokinesis, an orthokinesis, a tropotaxis, or a clinotaxis, you can set up a kind of a Monte Carlo type simulation to describe the movement of some particle in response to some attractant source at the center. And all of those can give rise to that same sort of that overall kind of macroscopic behavior where you see the aggregation of the population at a point source. So based on that is, if we start to kind of try and build up a model from this perspective of an individual, incorporating these different subtleties in terms of the way an organism responds to the environment, do you see a similar flux at the macroscopic level? Okay. Okay, so that's kind of our first motivation. Now, another motivation for trying to kind of start and build up from the level of an individual is also from a basic one of being able to connect the sort of data that we're able to obtain in a biological problem. So in terms of that problem on turtle navigation that I talked about, you can collect data at a macroscopic or a population level. So this is the arrival times of the female turtles that are coming back to the nesting beaches. They basically just, they basically have volunteers out on the beach that calculate the number, all of those female turtles arriving on the beach, and that kind of gives you some sort of distribution. And this is very much a kind of a population level here. We've kind of blurred all of that population into a single measure describing this arrival time. So that's kind of the macroscopic population level data that we obtain. But of course, then at an individual level, if we're kind of trying to follow the movements of the turtles as they get to the island, then you do that through fitting them with GPS devices, and that contains data on their movement speeds, their turning rates, etc., etc. So we'd like to be able to connect that microscopic individual level data to the kind of the macroscopic data in terms of arrival times. A similar thing can be kind of talked about in the context of cancers. So again, in one of those case studies, that kind of glioma growth, basically, the sort of macroscopic data is something like the distribution of a cancer as predicted by some sort of imaging, such as MRI or CT type scans, basically. That gives me a macroscopic distribution of that population of cancer cells. But if we go down to the individual level of movement and how they're responding to the environment, we can track individual cells and calculate their speeds, their turning rates, and how they're responding to their environmental information. Okay, so that's another motivation for trying to now connect from individual to macroscopic. So here's our kind of questions then. One, can we connect individual level measurements such as speeds, turning rates, turning distributions to the terms and parameters of a macroscopic model, such as diffusion coefficients? Under what assumptions do those classical continuous models, such as the Keller-Segel model, result from some underlying individual level description? So when can we obtain those models? What, can we get kind of a precise, sort of more precise information on that? And then a third question is, how does changing the behavior at the individual level influence the type of equation we get at the population level? If we change our individual from being able to do a clinokinesis response to a clinotaxis type, type of response, how does that change the sort of macroscopic model that we obtain? And all of these really kind of come around the central question is, if we begin with a random walk type model of movement, what sort of macroscopic model are we going to derive? And that's kind of the central question. And as I did mention yesterday, as soon as I've kind of tidied up these notes and corrected any sort of final sort of typos that I spot, I'm happy to distribute these to the conference organizers so that you can kind of get, if you want to kind of get some of the details on these slides. Okay, so this kind of question of being able to connect individual to connect a random walk model to a kind of a partial differential equation is because of a kind of old problem that's been studied by lots of people over the years. And that whole problem of the random walk was first proposed by Carl Pearson in a letter to Nature in 1905. There were lots and lots of studies, but probably from the perspective of biology, the key paper was that by Patlack in the 1950s. There are actually two papers on this, but really only one is really kind of widely cited. And that was this study in 1953, random walk with persistence and external bias, where basically he posed the question is, if you have a particle that is moving as a random walk through the space, there's some external bias that influences its movement in a particular direction. And moreover, that individual has a tendency to persist in a particular direction, which is, again, something quite relevant to a biological organism. What sort of macroscopic model will we get from this? And because this is such a kind of a key influential paper, if you look into the literature on chemotaxis equations, you'll often see Keller-Segel equations referred to as Patlack Keller-Segel equations. And this is actually despite the fact that actually in this original paper of Patlack, he never actually refers explicitly to chemotaxis, though it is something he clearly had in mind. So that was kind of the key paper in the 50s. And then really, in terms of kind of the more modern literature, that really kind of then sort of really kind of expanded in the 1980s with key works of Alt, Othmer, and Stevens and Hillens and other, where they were really kind of then targeting some of these kind of ideas towards this problem of chemosensitive movement. And I'll give a kind of a list of references at the end of today's talk, basically, at least of these key early ones, basically. Okay, so that's kind of the question that we're going to be looking at across the course of today's lectures. So let's kind of just set some sort of basic terminology. So I'm going to use the following. I'm going to denote xt as my random variable for a particle to be at some position x at time t. And for this particle, we've got a certain number of individual level parameters. We may know its speed, we may know its turning rate, we may know its turning distribution, etc., etc. These are all things that, as I say, if we're tracking a cell with a video, we kind of got some sort of experimental setup where we're tracking a cell, we can of course calculate these sort of these statistics, its mean speed, its mean turning rate, its kind of turning angles when it's kind of changing its direction and so forth. Those are the kind of the individual parameters. And then the sort of things that we would want to know at a population level would be very much these kind of statistical quantities, such as the expected position of the particle at some time, the variance in that sort of position, so how likely it's going to be distributed around that. We'd be interested in knowing the density distribution of some population of individuals. And we could also maybe be interested in the mean time to hit some target. You know, that would be relevant for my turtles trying to get back to Ascension Island, or an immune cell being able to hunt down some infection, etc. And then possibly various others as well. Okay, so we want to try and connect between these two, from these individual parameters to these population measures. And I'm going to go through, today's lectures are going to be very repetitive, basically, because we're going to go through and derive effectively the same equations again and again. But we're going to do that through a step-by-step process, where we're going to start with our simplest case, where we just take a very simple uncorrelated random walk on a 1D lattice. And the calculations here are going to be very, very straightforward. We're then going to talk about an uncorrelated random walk on a 1D lattice, but with a control species. We're then going to talk about a correlated random walk on a 1D lattice. So this will all be my kind of one-dimensional part of today's lecture. Then we're going to move up into higher dimensions, and we're going to talk about two general models there. We're going to describe what we call a position jump random walk in ND. And we're then going to talk about a general velocity jump random walk in ND. And I'm going to try and do this in a way where hopefully the calculations are all going to be relatively straightforward ones that you should be able to kind of follow quite straightforwardly just by kind of following through the slides. But again, if you want more information on these, then as I say, I'll kind of be adding kind of various references at the end where you can kind of find more fundamental calculations here. Okay, so that's the kind of the plan of action for the rest of today's lecture. So as I say, let's start with the simplest case, and that's going to be of my simple uncorrelated random walk on a one-dimensional lattice. So this is the very simple setup. We have a particle that starts at position x is equal to 0, and then we suppose that in each time step of duration tau, it either jumps delta to the right with probability q, or it jumps delta to the left with probability 1 minus q. So we're already imposing that it has to do a jump every time step. So here's the kind of the picture of that. It can only move along this one-dimensional lattice. It can only move in steps of length delta. Starting from some position, it will jump right with probability q. It will jump left with probability 1 minus q. Okay. This is about as simple as we can frame this sort of problem. Well, let's then, as I say, let's kind of denote by xn to be the random variable for the position of my particle after n time steps. So that's kind of my random variable. And the first question I could kind of try and come up with, if I'm trying to get those kind of population those population sort of measures, is what is the expected position of the particle after these n time steps? Well, this is actually a very straightforward calculation. If we want to calculate the expected position, then we just sum the product of the possible locations with the probability of it being in that location. So we could do this very easily for the expected position after one time step, the expected position after one time step. Well, we start at 0, so it's either going to be at positions delta or minus delta. It'll be at position delta with probability q. It will be at position minus delta with probability 1 minus q. So the expected position after one time step would be simply delta q plus minus delta times 1 minus q, and that's delta times 2q minus 1. So incredibly straightforward calculation. That was very easy. Let's do this for 2 time steps. Well, in this case, after 2 time steps, you're jumping every position. So now you've got 3 positions that you could be in. You could either be at position 2 delta, you've done 2 right jumps. You could either be at position minus 2 delta, you've done 2 left jumps. But of course, you could have jumped right, then left, or left, then right. in which case you're going to be back at the origin. So expected position after 2 time steps, you do a similar sort of calculation, gets a little bit longer, but it simplifies this down, and you get simply 2 delta times 2q minus 1. So again, it's a very, very straightforward calculation. And if you go to the third time step, of course, that calculation is going to get a little bit longer, because then you could be either at 3 delta, you could be at delta minus 3 delta. There's going to be more steps along the way, but you could actually do this a lot more quickly by simply kind of noting that there's a kind of an induction process going on here, because actually if we kind of go back to that expected position after 2 time steps, well, it's simply going to be your expected position after 1 time step plus another movement right or left. So that will get you to exactly the same calculation. And then by induction, that allows me to then very quickly write down the expected location after n time steps. It's going to be simply n delta times 2q minus 1. And this is already then giving me a picture of how that expectation will depend on the parameters of individual movement. It's depending on that kind of that step length delta, and it's depending on that probability that's determining whether or not you're moving right or left. Okay. Now, we can also do these calculations for the variance. The variance, again, it's again, slightly more complicated, but these are all still very trivial calculations. We just compute using the standard formula for the variance. And you can compute that for after the first time step. You can compute a formula for the variance, which will be 4 delta squared q1 minus q. You can use induction again to compute the variance after n time steps, and you get a formula such as this. Okay, so for this kind of random walk process, as I say, this very simple one, in just 2 or 3 very, very trivial algebraic calculations, we've got formulas for the expected location and the variance after some n time steps. So let's put that together. Those are kind of my expected locations and my variances. And what I can kind of see from this is, well, if I set that q to be equal to 1/2, then my probability of moving right or left is going to be the same. And then actually, as we would hope and expect, the expectation vanishes. You're equally likely to move right or left. So the expected location will simply be 0. And that gives rise to what we call an unbiased random walk. If q is greater than 1/2, then of course the expected location is now going to be to the right. We're now going to get a kind of a general movement towards the right. And similarly, if it's less than 1/2, that expected location is going to be less than 0. So we've had this kind of general movement to the left. And then as kind of q tends to 0 or q tends to 1, then in that case, basically, we get into the situation where the particle just always chooses the same direction. And in that case, my variance sort of vanishes, and we're just simply moving straight to the right or straight to the left. So you can get these sort of basic sort of properties then of how my particles are moving. And to give some sort of terminology here, let's now kind of start relating this to a kind of actual kind of space and time scales. We had our particle moving a step tau every time step. So after n steps, we can define a time t, which is simply n times the tau. And we can calculate the mean velocity of my particle. That will be simply the expected location after n time steps divided by t. And that gives me a quantity delta 2q minus 1 over tau. And we can describe a diffusion constant for this in terms of the variance. And that gives me this formula here. But basically, as I say, what we've done here then is, in this very simple context of a random walk moving right or left on a lattice, we've got our kind of first insight into how macroscopic parameters such as mean velocities and diffusion constants relate to those underlying parameters describing that random walk process. Okay. Okay, so that's my simple position jump random walk in one dimension. What else can we do with this? Well, we can also try and get an equation for the probability of a particle being at position x after time t. So now we're going to define P of xt to be that probability of a particle being at xt, and I get an equation for this. And in what follows, I'm going to assume that my kind of random walkers, I've got some population of independent walkers. And in that case, we can kind of reinterpret this as effectively the density of a population of independent walkers. But you can think of it in those two different ways, either the probability of being a single particle at being at position x at time t, or if we have a population of independent walkers, we can think of it, regard it as a density of that population. Okay. So how do we do this? Well, we've got the same underlying random walk process. And the first step is to describe the discrete space, discrete time master equation. If we were, my particle was at position x at time t, then we want to compute the probability of it being at position x at times t plus tau. And of course, that's going to be the probability of it having been at position x minus delta at t and jumping right. So q times p, x minus delta t, plus the probability of it having been at x plus delta at time t and jumping left. And that gives me my discrete space, discrete time master equation for this process. You're either, in order to be at position x at times t plus tau, you had to have been either at x minus delta and then jumping right, or at x plus delta and jumping left. So that's my discrete space, discrete time master equation. So how can we move this up into an equation for P of x at t? Well, we can do expansions about xt. You know, these are all kind of, this is P of x at t plus tau. So we can expand as a Taylor expansion about xt. We can expand these as Taylor expansions about xt. I'll drop the xt dependence on the right-hand side as follows, but just applying standard expansions, that expansion for this left-hand side here, you expand it, around that sort of point there, and you get kind of your standard Taylor's expansion. Kind of the right-hand side, we expand each of those, etc. And we're going to then substitute those into that discrete space, discrete time master equation. Okay, so we take those in, we substitute. You'll find that various things nicely cancel each other out. And then when you divide through by tau, you get something that's beginning to look like a partial differential equation. But there's still clearly a lot more mess here. On this kind of right-hand side, we've got a kind of this kind of first term here is in terms of dp by dx. The second term is in d2p by dx squared. Of course, we're going to get various other higher order terms in there as well. And the question we ask at this point is then, from this, what sort of limiting equation can we obtain from this? So substitute and rearrange, we get to this, and we then look at the limiting equation. Well, here's where we then have to be a little bit careful, because the sort of limiting equation that we get is clearly going to depend on the size of Q. In front of the dp by dx, we've got a delta over tau, but we've also got this 2q minus 1. In front of the d2p by dx squared, we've got a delta squared over tau. So we're going to have to be a little bit careful about how we get our limiting equation according to the sizes of these various terms. So let's first suppose that this kind of 2q minus 1 is of order 1. Well, in that case there, we're going to let my delta and tau tend to 0. And we're going to do that in a way such that the limit of delta over tau as they tend to 0 is going to be equal to some constant. And then, well, we can see what we're going to get. That's maybe going to change sort of the pen in this sort of case there. If we kind of assume this, if we assume this here, then this term will disappear, and all of these terms will disappear as well. And then that kind of then tells, when we kind of take that limit, we get a drift, a simple advection process here. It's a drift dominated equation. dp by dt plus c dp by dx is equal to 0. Okay, so if we've got this sort of situation, we get a standard advection equation in that when we take the limit. Okay. But what about if instead 2q minus 1 is around order delta? Well, if that's the case, we're going to have to do a slightly different sort of scaling. We do what we call a parabolic scaling. We let delta and tau tends to 0, such that the limit of delta squared over tau is equal to d, and the limit of delta 2q minus 1 over tau is equal to c. Because we've got this, that 2q minus 1 is of order delta there, then these are both going to be a similar order. These will all disappear as before. And then in this sort of equation, in this sort of limit, we get a drift diffusion equation. So that's my drift diffusion approximation. And finally, we can look at the case where 2q minus 1 is order delta squared, or even less, even potentially 0 here. Well, in that case, again, we take this kind of parabolic scaling. We set the limit as delta over tau, of delta squared over tau to be equal to d. D is equal to constant. In this case here, this term will disappear, this term will disappear, and we get left with a diffusion equation. So the type of limit and the type of equation that we get will crucially depend on the size of delta. Okay. Okay, and that's, as I say, my diffusion dominated case. Okay, so you can also use this same method to obtain other sort of measures at a population level. So similar methods can be employed also to solve what we would call first passage time problems. So I've been talking about my kind of turtles getting back to an island here. This case of getting back to Ascension Island. And for example, this is a study of Hayes in 2003, where basically they released turtles either northwest or southeast of the island, and then tracked their movements as they were trying to get back to the island. And what they actually found out was that 4 days later, only those that had begun northwest, so these ones that were kind of up here northwest of the goal, had returned. The ones southeast of the goal hadn't managed to get back. And this was actually very much, this was, they kind of speculated, was very much down to kind of the patterns of wind around the island, but I'm not really kind of going to go into that here. But this is essentially what we would call a first passage time problem. We're taking some population, we're releasing them, and then we're interested in the question of how quickly they're going to be able to get back to their goal. So you can also kind of use this sort of framework to kind of set up and solve first passage time problems, because if we take my random walk on a lattice, we have my individuals jumping left and right. Then if we define my call it t of x, to be the mean first passage time from a position x, then we can write a discrete space, discrete time master equation for t of x. And basically, this is this is kind of now in time. And what this basically says is that the mean first passage time from position x is going to be a kind of a time step tau, because in each step takes place after tau, after an interval of tau in time, plus the mean first passage time for having been at x minus delta and jumping right, plus the mean first passage time from position x plus delta and jumping left. So that gives me a discrete space, discrete time master equation for the mean first passage time. And then I could do the same sort of thing. I can expand about x, and I go through this much sort of quickly there. You kind of go through those expansions. You do the kind of standard sort of tidying up. You do your kind of scaling sort of process. And in this case here, taking one of those particular equations, and it gives rise to that equation, an equation which is essentially of the form of a second order partial differential equation for your mean first passage time. And then in order to solve a particular mean first passage time, you would set that up and solve it according to appropriate boundary conditions. So if we're talking about this on a one-dimensional line, maybe you have your target at position x is equal to 0. And if that's your target, then that immediately says that the mean first passage time point from 0 has to be equal to 0. You're already at the target, so your mean first passage time from there will be equal to 0. At x is equal to L, you just say that you've got no loss. You put an appropriate no loss condition there. That gives you a second order differential equation with two conditions. You can solve that and get an explicit equation for that. So this kind of general approach, basically, where you take that random walk, you write down the discrete space, discrete time master equation for that, you take expansions, and then you look at, you kind of take an appropriate kind of scaling limit of that, allows you to go from these underlying kind of properties of your random walk process to macroscopic measures, either population densities or, as in this case, a mean first passage time. Okay. Okay, so how can we expand this? And as I say, we're going to go through effectively the same things time and time again. Let's now make this slightly more complicated. So only marginally more complicated. We assume basically again that we've got a kind of a particle jumping right or left on a lattice. It's always jumping a distance delta. Now we're going to assume that that it kind of jumps to the right with probability q plus x and to the left with probability q minus x. And of course, these probabilities would need to be appropriately bounded because we are dealing with probabilities there. So, you would need kind of appropriate bounds on them. I'm going to kind of ignore that kind of issue here in what follows. We're just going to assume that they're going to be appropriately bounded. But we're now going to assume that these are potentially time dependent. So we write down the discrete space, discrete time master equation in this case here. And it looks somewhat similar. There's a slight sort of difference here because now when we kind of write that down here, then we write down the kind of the probability of it being a position x at time t plus tau. And we're going to have terms on the right-hand side where you would have either jumped right from position x minus delta, you would jump left from position x plus delta. But now, because of course, it's not necessarily the case that these q minus x plus q plus of x sum to 1. So there's also now a contribution from the probability of actually staying at position x. So you've now got 3 terms on the right-hand side. Okay, so, but apart from that, it's essentially the same as previously. And in particular, for what we want to do, we're going to assume that these Q plus X and Q minus X depend on some external control species, call it C of XT. And in the context of chemosensitive movement, this of course could be a chemical concentration if we're trying to describe something like chemotaxis or chemokinesis. We're making that probability of a movement now depend on the local environment that some chemical species C, which is going to be a function of potentially X and space and time. Okay, so that's our kind of now extension to include a dependence on some control species. And we can start to then set up different models for how we move from one position to another. And I'm going to assume just in what follows that we just base our decision on 2 locations. Potentially, we could actually set it up so that actually it detects C over the full region of space. But we're actually just going to assume that my decision to move depends on information at my current site and information at my target site. So we're going to just look at these 2 pieces of these 2 positions and make a jump accordingly. But we can do this in various ways. So, okay, sorry. So Q plus or minus is a function of C at my target site and C at my current site. So in the first case, I could consider what I call a strictly local or myopic walker. And in this case here, you make that decision to jump depend only on the value of C that you're seeing where you are. You look down, you look at what C is at your site, and you jump accordingly. And of course, at that point, whether or not you jump right or left, it's going to be the same. You don't have any information to tell you which way you're going to move. So Q plus of X is going to be Q minus of X. And as a prototype model for this, I'll just simply take this to be alpha times C of X at T. So you're more likely to jump the higher C is. Okay, and that's what we would call a strictly local or myopic walker. You just base your decision of whether to jump on the amount of C at your current location. You could also set up a destination dependent where it depends only on C at the target site. So you look at the target, you look at the amount of C there, and well, in the case of Q plus of X, then you're going to say alpha, you're going to jump there according to the size of alpha C at X plus delta, and vice versa for jumping left. That would be a destination dependent case.
Kevin. There's a question.
Yeah.
Can you hear me?
Repeat the last little bit. Okay. Yeah. So.
My question is actually in the previous slide, I guess, for the first, so the mean passage time for the first.
Oh yeah.
Yeah. I'm not sure if I got that right, but then this first time means that we go from one position to the other, right?
The mean first passage time is the average time that it would take starting from some position of space to reach a particular target, basically. So, so think of your kind of turtle homing problem in this sort of case there. Then you have a target with it, which is the island. You can start at some position away from that. You do a random walk, and starting from there, there's going to be some mean time that it takes before you reach that target. That's what the mean first passage time is.
Okay, then what does tau mean in the equation?
So tau is, whoops, okay, let's go back to there. So in this equation here, tau is your time step. So because this is now an equation for time, what's happening in this equation is that basically, you're kind of writing an equation that said if you want to write down an equation for your mean first passage time from position X, then that's going to basically, that you can explicitly link that to an equation for the mean first passage time from positions X minus delta and X plus delta. So, that, and of course, from if you were to X minus delta, then you would have jumped right with position with probability Q. If you were to X plus delta, you would jump left with probability 1 minus Q. But of course, this has also taken an amount tau of time. So we need to add that tau to this equation as well. Okay.
Okay, but then if we take the case of the sea turtles, then the sea turtles will take the same amount of time to go left and right. This is right?
I mean, in this very simple sort of case here, we're kind of assuming a very simplistic sort of random walk. This is not really, this is more just to give you a motivation of why we could be interested in a mean first passage time problem, rather than actually this being a specific model for sea turtles. This is just a kind of a motivating case. Okay. Okay.
Well, maybe I also take the chance to ask a quick question. Like in the first time and space dependent case for the Qx plus 1, Qx minus 1, like, are we still assuming that there are probabilities in the sense that they have to sum up to 1, so there is also probability to stay at the same location, or? So in this case here, where we now kind of have them as Q plus X and Q minus X, yeah? Yeah. No. In the next slide, I think, like the first time dependent case. So what was this?
No, now they can act Q plus X plus Q minus X can basically be less than 1, basically. So formally you would kind of have to sort of set down certain, maybe as I say, maybe I should just write this down quickly. Okay. So what we kind of need as a kind of a set of assumptions is, my Q plus of X, it's a probability. It's got to be between 0 and 1. Q minus X has got to be between 0 and 1. And similarly, kind of the sum of those has to be between 0 and 1 as well. So those are the actual, that's what I meant by appropriately bounded. But what this does, of course, mean is that now you could potentially stay at your position as well, basically. You don't necessarily have to jump. So you could stay there, and that's what we're kind of assuming. Okay.
I understand. Yeah, thank you. It was more for this, the first example that you have in the next slide, where the Qx plus and the Qx minus are time dependent, but they are equal. So it means that then there is a positive probability.
Yeah. Formally, this of course sort of imposes some conditions on the size of C, basically. I'm just trying to put down the simplest possible model here in order to keep all of the kind of calculations as easy as possible. But if you want to kind of go through formally, you should put in those kind of the appropriate bounds and make sure these functions are bounded appropriately and so forth. But we're just going to implicitly assume that. Okay. 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 So as I say, we've got these, so I think, so here is where I was at. So we've got different ways that we could choose to jump right or left. We could have a strictly local myopic water. So you only base your information on your current site. You could have a destination dependent where your probability to jump depends only on the information at the target site. You could do some sort of an averaging process as well. You could basically look at your current site and the target site and then average the information across those two locations. And you could also do a comparison sort of process. So something like where you basically look at the difference in C at the current and the target site here. And this is actually not all of the different ways that you could kind of model this, basically, but they're kind of 4 models that basically allow you to balance information between your current and target site. And this kind of comes from kind of a lot of this kind of really comes from a kind of a study of Othmer and Stevens in the late 90s here, basically. Okay, so those are my 4 different models. And as I say, as soon as we've finished this kind of control species case, we're going to have the break, because I realize we're kind of running up almost to 2 o'clock now. So let's take the strictly local case. And again, we just assume the simplest possible one. We set Q plus X is equal to Q minus X is equal to alpha C. Well, you do the same process, and I won't go through these calculations in detail. It's just really, very, very standard Taylor expansions and then applying these various sort of limits there. You kind of write though, you kind of put that into your master equation. Of course, it now looks a little bit more complicated on there. You do exactly though as before, you expand about xt and you take a limit, and in this case, you can take a kind of a parabolic limit, such that limit of delta squared over tau is equal to d alpha. Oh, sorry, there's actually a typo here actually there, so sorry about this. I've just suddenly seen there. There should be the limit of alpha delta squared over tau is equal to d. Okay, so you take that sort of limit, and then in this case, in my macroscopic model, we now get we get a kind of a macroscopic equation which depends on both C and P, and expanding it out, I can run and write it in this sort of form here. And one of these terms is a diffusion type term here. We've got kind of a diffusion term, but it depends on the size of C. And this other term here is describing a taxis type term, but in this case, we've got kind of a plus there, and that's describing a drift down the gradient of C. But in other words, that's the kind of the first hint of being able to derive a taxis type equation. Okay. Okay, so, but anyway, apart from that, it's essentially the same calculations. Now, what about, sorry, and let's just to kind of note here, this could be thought of in a way as being a chemokinesis type sort of behavior. We're only looking at the concentration where we're at, basically. There's no gradient sensing at all, where you're kind of looking at the gradient between sort of 2 sites. You're just detecting a concentration. So this is perhaps describing something that looks maybe chemokinesis type. But that was only one of my models for how I could kind of jump from right to left, because it could be that my particle decides instead to look at the destination. So Q plus minus of X is now equal to alpha C of X plus minus delta T. You look at C at the destination. And can anybody think about what we're going to get in the limit? Does anybody want to even kind of tell me what they think we're going to get if we go through the same process?
Yes, we will get 2 times derivative of C times derivative of P plus C times the Laplacian of C.
Mm-hmm. You go through this.
No, okay, no, it was a big deal.
Okay, it was worth a guess. It looks very similar what we get, but actually now we've got a crucial sign difference going on in here as well. Whoops. So again, we've got a kind of a diffusion type term, and this is a taxis type term, but up gradients in C. Okay, so that last one was a kind of a taxis down the gradients in C. Now we've got a gradient, a taxis up the gradients in C. Okay, so, but again, essentially, we've shown again how you can you can go through the process. What about averaging? What will I get in this case? Anybody want to kind of guess it in this case?
No gradient.
You mean kind of no taxes part, yeah? Yeah, exactly. You can actually work this out because it's going to be the average of these 2 models here. Those terms are going to cancel with each other, and then you get just simply a straightforward diffusion process. Okay, so you get, again, something similar-ish, but not quite the same. Okay. And finally, in a gradient sort of case, or in this case here, you compare the gradients across the two sort of models. And this is actually now where you get, well, really, in a sense, the first thing that really looks like one of the models. Oh, actually, they all looked pretty well like the models yesterday. But this is actually now directly related to one of the models I introduced yesterday. This is now really your minimal Keller-Segel type model. You've got diffusion and you've got taxis going on. But the point is, with this, yeah, we've kind of assumed a lot of similarities. We've assumed that you can just kind of move according to information at either your target site or your destination. We've assumed subtle distinctions between the two. And we get similar sort of macroscopic equations, but we get some slight differences going on. And you can kind of think what will happen if you start with a kind of a C, which is kind of distributed, you know, like a kind of a pipette releasing an attractant. So kind of concentrated, you know, as a kind of a source that spreads out from some location. In this case here, my population would move down the gradients. So after time, it would, you know, it would be accumulated away from that peak. In this case here, it would kind of move up. In this case here, of course, it's just a standard diffusion process, so we don't get any accumulation of the particle. And in this case here, you know, again, we would accumulate up, basically. So those subtly distinct rules at the microscopic level will influence the type of behavior we see at the macroscopic level. Okay, and you know, you can kind of, I mean, not in a kind of not in an absolutely, you know, sort of precise way with this sort of very simple model, but you can kind of talk about these kind of, some of these models as being chemokinesis type. This last one's more of a chemotaxis type one because you're biasing your movement according to an actual local gradient in the attractant there. So it kind of relates to those underlying microscopic models. Okay, so I think actually at that point here, we're kind of stopped for sort of about maybe sort of 5 minutes or so everybody can have a kind of a quick sort of break and a refresher, and then kind of continue with the kind of the next part of today's course. I won't get through absolutely all the way through to velocity jump random walks today, but we will get to that then in Thursday's lecture. But we'll finish there for about 5 minutes and pick up again in another then with the second half of today's class.